Good morning, everyone. I thought we'd start promptly. Sorry, it's 8 a.m. We get that awesome time. And I am really excited to introduce a diversity of speakers today. And in general, in the work that we do in mapping, mapping can be at any scale, and whether that scale is local or global, and then also scale with whether that's at the species level or at the landscape level. And we've got a really exciting lineup of speakers, a really neat mix of folks from academia to on-the-ground restoration, and you'll see a variety of standards and techniques and a variety of work that people are doing, whether that's to produce vegetation maps for on-the-ground conservation and restoration planning, or otherwise utilizing geographic information systems and new and tried and true technologies, including LiDAR, remote sensing, and a variety of analysis techniques. So we're really excited. I think you've read the session title, and that's why you're here, so I'm not going to read it all. I just wanted to introduce my co-chair, Michèle Slaton, from the U.S. Forest Service, and I am Julie Evans from the Vegetation Program of the California Native Plant Society. So the first speaker I would like to introduce is Israel Borokini, and he is a postdoctoral scholar at the Department of Integrative Biology of the University of California, Berkeley, and he has completed his PhD in ecology and evolution and conservation biology at the University of Nevada, Reno, and his research interests include plant ecology and biogeography, and spatial phylogenetics, and landscape genetics. So we're really excited to have you.
Thank you very much, Julie, and thanks so much for being here, for waking up at 6:30, 7 o'clock to be here. And I just driving about an hour from Berkeley to come here as well. And also, before I start, I just wanted to give a shout out to my mentor, Brent Michèle, who is the professor and the director of University and Jepson Herbaria at UC Berkeley. So I'll be talking about the use of spatial phylogenetics for conservation-driven land management in California. This is a relatively new and developing set of tools and metrics that could be used for mapping diversity across in a particular area, which is not bounded by any boundary. So what is spatial phylogenetics? Basically, spatial phylogenetics is like placing the tree of life or any phylogenetic tree on a map and just seeing which areas have unique biodiversity given the phylogenetic or the evolutionary relationships and which areas have less. And so that is a layman's way of explaining it. Basically, what do you need for that? You need a phylogenetic tree, obviously, or the tree of life, and you need a set of coordinates or occurrence records of the particular taxa you are working with. And of course, the data, the taxa you're working with must match for both the occurrence and for the tree. It can be applied at any taxonomic level or geographic scale. In fact, this is a time when paradigm is shifting from species-focused conservation to land-focused conservation, because up till now, habitat loss and destruction remains the greatest threat to biodiversity at any scale. And we don't have all the expertise and the money to study every individual species in the world, and some of them might be lost before we even get to study them. And so it is really important to shift that paradigm from species-centric conservation to land management and land and habitat management. And this method underscores that focus. And when we talk about, anyway, I'll come back to species later on, but what spatial phylogenetics measure is it measures phylogenetic diversity and endemism based on branch lengths. How long or short are the branch lengths, and how related are they on the phylogeny? It is rank-free, which means it can be done at genus level, family level, species level, below species level. So it doesn't necessarily have to be at a particular rank. And what makes it really, really helpful now is because we have a huge, we have technological advancement, which makes a lot of data available now. We have the GBIF, we have the CCGP going on in California. We have several other projects whereby biodiversity data is being made available. And we also have computer development, which also makes it possible to be able to work with big data sets without having to use a local machine, which currently right now have an analysis has been running since Saturday, and it's not yet finished running on my local computer. But you can do that. We don't have to be limited by that. And spatial phylogenetics has a lot of application in different fields of study. So talking about biodiversity is the sum total of all plants, animals, fungi, and microorganisms on Earth, their genetic and phenotypic variation, and the communities and ecosystem of which they are part. And so when we talk about species, we are talking about just one tiny aspect of biodiversity because there's so much to biodiversity than just the species. And this is really, really important. Of course, to the general public, species is still important, but just think about it in terms of statistics. When we have things, numbers like 1.1, 1.2, 1.5, 1.7, 1.8, and so on and so forth, and then you just sum everything up and you find the average, and you use the average for whatever you want to use it for. That average number that you are using, or the mean value you are using, you have lost a lot of information that it is representing, and that is where species comes in. So when we group some bunch of populations and we say, yeah, all these populations, cluster them into one species arbitrarily, we have lost a lot of information that is below species level for those particular groups of individuals. And this is where spatial phylogenetics or evolutionary methods are really, really important, complementary to what we have currently on mapping biodiversity. For the sake of time, I'm just going to move forward. So what's a phylogeny? So I'm just going to go through the anatomy of a tree very quickly. If you already know this, my apologies for insulting your intelligence, but I'm just going to make it very quick, I promise. So, of course, we have what we call the terminals or the tips, and that's where we could say it could be family, it could be genus, it could be species, it could be populations, it could be anything. And then we have the branch length. The branch length represents the amount of evolutionary change or mutations. I mean, we've all taken biology classes whereby nucleotide sequences change. Adenine is replaced by cytosine, or thymine is replaced by guanine, and so on and so forth. So those changes along in nucleotides are and over time, is what is reflected in the branch length. And those branch lengths, and then we have parents and child. That is where the nodes comes in. The parents and children, what give rise to what, up to the end. And then we also have, because it is constructed using statistical methods, there has to be some bootstrapping and there has to be some measure of support. Think of it as like p-values, but not necessarily p-value, but something to tell you whether there is strong confidence in that split there. And then to the last common ancestor. And then we have a scale which is used to actually calculate the branch length. And this is really important because most of the spatial phylogenetics metrics are based on the branch length. We're going to be using the branch length a lot in those estimations. So we could have 14 taxa or species, if you want to call it that way, but all of them are clustered on one part of the tree, which means they share so much evolutionary relationships, and there's not a lot of diversity among them compared to the other tree, to the tree on the right side that captures different aspects of evolutionary relationships and uniqueness across the tree, even though they are both 14 terminal clades or taxa on the tree. So this is where phylogenetic diversity comes in, which was developed by Daniel Faith, which is defined as the sum of the branch lengths that connect all the terminals to the very root of the tree. So so using the branch lengths, the estimation of the branch length, you just simply sum them, 0.1, 0.5, and so on and so forth, till you get to the very root of the tree. And so this is another classical way of looking at it, whereby you can say, well, we have 4 taxa in that grid cell. Where does space comes in? Like we're talking about spatial phylogenetics. So we have 4 taxa on the grid cell. Those 4 taxa are found in this area on those portions of the tree. We can count and we can add it up and to the very root of the tree to be able to estimate the phylogenetic diversity of that particular grid cell. This is where space or GIS comes in. And we can say, okay, well, this grid cell has a lower phylogenetic diversity compared to the other grid cells. So this is the logic there. And again, this is another way of looking at it using another tree, whereby we can say, well, those blue filled circles, they are all found in one grid cell. We can count, estimate the PD for those, and we can do the same for another set of 3 taxa and so on, and calculate the PD for different taxa in different grid cells. And then the second, because endemism is really important, because oftentimes we look at taxa that have limited range or they are range restricted, often are given higher priority for conservation. And so there's a way to also estimate phylogenetic endemism, whereby the PD is divided or weighted by the range of that tree, of each taxa in the grid cell across the entire study area. And that is what is called phylogenetic endemism. And so basically you just estimate for each taxa how many places or how many grid cells do they occur in. And then you divide it, and you divide the PD by the branch length by the number of grid cells where they occur, and then you also estimate the PE. And so by doing that, you're able to shrink some of them that are widely distributed and give more priority to range-restricted taxa. All right, I'm going to move faster now. So relative phylogenetic diversity. So PD is by default or by chance, the number of taxa you have on the tree will likely increase the number, the value of PD by default. But one way we can estimate whether the PD we discover or we found is actually give us a significant concentration of longer or shorter branch length is we can say, okay, well, this tree, let's give all of the branch lengths equal value. All of them, all the taxa have exactly the same value. That is a null tree. And then we divide the actual PD by the null tree, and that will give us a relative PD. And this is what we have here. And that way we can we can detect which areas have a significant number of longer branch lengths. And what do we mean by longer branch length? It means that they are phylogenetically older taxa. They have accumulated a lot of ecological or geological history through time, and they are really, really important for us to study those kind of taxa to know, okay, if these taxa they have a lot of accumulated history, we can use information from those taxa, functional traits or whatnot, to predict what kind of taxa are going to survive climate change or what is going to happen next. And then similar logic for phylogenetic, for relative phylogenetic endemism. But here the problem is when we do that, it gives us such a calculation that it gives higher preference for longer branch lengths. But there are some other taxa that are shorter branch lengths and also has restricted range sizes. Those are not given preference, and this is where we have canopy comes in, which I will explain later. Now, bringing that into California, how can we use this data or this set of tools to determine which areas need to be protected in California? And of course, we all know the uniqueness of Californian biodiversity. Because of time, I won't go into details of that. And so how do we do this is we have herbarium records, and this is where giving a talk like this in this session is really important, because when we all go out to do vegetation mapping or to do collections, it is really important to collect voucher specimens, because those voucher specimens are critical to providing reference information that can be used for several other things that goes beyond just the mapping itself. And so please, as much as you have the opportunity and the ability, please collect voucher specimens, and we are happy to save them for you, or you can even develop your own herbarium in your own house. And so we have herbarium records. We can fit niche models to predict the range, or we can just use those raw values and bring in the phylogeny, and we can use that to predict areas that are important for prioritization. I will go quickly, so I won't waste time on that, take too much of time on that. These slides represent a study that has already been done, which was published in 2018 by Matthew Kling and several other co-authors at UC Berkeley, whereby they use about 1.4 million specimen records in California for Californian plants, and with the corresponding phylogeny to map areas that are important for conservation. And so using those information, we have this set of maps. The first one is TL, representing taxonomic richness, or what you call species richness. And then we have the weighted endemism richness divided by their range size. And then we have the PD and PE. You can see that the TR and PD are very tightly correlated. They have very similar patterns. And then we can also do randomization, whereby you can scatter things around and see, making sure that each grid cell has the same number of taxa originally that it has, but just moving them around randomization. And then it can tell us how significant, which areas have significant phylogenetic over-dispersion and clustering, and then we can also do the same for the RPD. And this showing us maps of areas, those ones in blue shows areas that have significant long branches and areas of significant short branches, and there's a lot of ecological and evolutionary interpretations to that. And then the canopy identifies areas that have endemic neo and paleo endemic species, as you see it on the slide. And we can also do phylogenetic beta diversity in terms of turnover. Of course, we all know turnover in community ecology. And we can also do exactly all of what I've explained so far. We can do them on 3 different types of phylogenetic trees. We can do them on a tree that is based on character divergence. We can do that on one that has to do with time, which is called chronogram, or one that has to do with just looking at rates of net diversification. And I'm going to stop here at this point out of respect for time, and I'll be happy to answer any question. There are still a couple of slides, but I'm just going to stop here right now.
Sounds like next time we'll need to give you 40-minute time slot. All right, we've got, it looks like 2 questions. We'll see if we can at least take one. Yes.
Since you've been cut short on time, where can we read more about this?
Yes.
Oh, thank you so much for that question. So This particular set of slides and presentation was based on the study that was conducted by Matthew Kling, and several other co-authors, including my mentor, Brent Michèle. So you can find those on the UC Jeps, the University and Jepson Herbaria website at UC Berkeley. If you Google Brent Michèle, you can also find a number of studies that have been done using similar approach. There is a particular software called Biodivers. The software is called Biodivers. It has all of these metrics in those, and there's one interesting thing about the software is that the developer also has a web page that listed all of the papers that have used those methods, and so you can read a number of papers on those. So the word Biodivers, that's the software. Thank you.
Yeah. That was amazing. Sorry that you couldn't show those last slides. They look amazing. Let's see. It might have gotten changed. Oh, goodness. I am gonna, slideshow. Sorry, it's very tiny here. Current slide. There was a lot more here. I had to show it. Okay, great. All right. Our next talk is by Christopher Kibler, and he is a PhD candidate in the Department of Geography at the University of California, Santa Barbara. And his research combines remote sensing and field ecology to study how plants respond to fires, droughts, excuse me, and other disturbance events. And he is currently a NASA future investigator in Earth and space science technology. So, welcome.
Hi, everyone. My name is Christopher Kibler. I'm a PhD candidate at the University of California, Santa Barbara. Today, I'm going to be talking to you about our research examining post-fire vegetation recovery during extreme drought. I'd like to thank my collaborators, Anne-Marie Parkinson, Dar Roberts, Carla D'Antonio, and Seth Peterson. We also had two very hardworking field technicians, Monica Goodwin and Peyton Thomas. We'd also like to thank the Forest Service, who funded this research, and NIFWIF, who administered the grant. And so, as you all know, as a result of anthropogenic climate change, we're seeing more frequent and more severe droughts here in California, it seems, every single year. And as the landscape becomes drier, it also becomes more flammable. And so one of the side effects of that is we're also seeing bigger and more frequent wildfires across the state. And one thing that's happening because of that is that when you have a fire that moves through a landscape, there's an increasing likelihood that that post-fire recovery is going to happen during drought conditions. And so you have these compound disturbances where the wildfire affects the vegetation, but then as the vegetation is trying to recover, it's dealing with this other disturbance in the form of drought. And these compound disturbances, I think, have really great potential to reshape native plant species here in California, both in terms of abundance and diversity. And that can happen at a really local scale. It can also happen at entire landscape scales. And so we think that it's really crucial to monitor these trends and these behaviors at both local and landscape scales. And that's what I'm going to be talking to you about today. We examined one fire for this study, and that was the Zaca fire, which burned in Santa Barbara County in 2007. At the time, it was one of the largest wildfires in the state. Quite unfortunately, that's a record that's been broken many times since then. But it burned about 97,000 hectares, which is 250,000 acres or so. And most of the burn scar is in Los Padres National Forest. And it's a really interesting burn scar to think about from a vegetation perspective because it covers a really large elevation gradient. And so, at the very lowest elevations, down maybe 250 meters, we see species like hard chaparral. And then as you move up through the landscape, you start to get into oak woodlands. And then up by 2,000 meters, you get some really nice sky island conifer forests in the San Rafael Wilderness. And another really interesting thing about this fire is that the fire itself burned in 2007, and there was 5 years of normal recovery, we'll call it. And then the really severe California drought hit in 2012. And so the recovery from this wildfire was very much affected by that extreme drought that I'm sure you're familiar with, and in a lot of ways is still continuing today. And so for this study, we considered two different methods that we thought were complementary, field surveys and remote sensing. Field surveys are great because they provide really reliable data about abundance and diversity across the landscape. You can send out a crew of trained technicians. They can measure individual plants. They can look at the composition of a stand, measure plant demographics and things like that. And so you get really reliable, really consistent measurements. But the downside is obviously that field surveys are very expensive. There are only so many places a field crew can visit in a season, and keeping that field crew staffed for longer than a season often just doesn't take place. And so you get really good data, but on kind of individual snapshots of the landscape. Remote sensing, on the other hand, can really be easily scaled across space and time. And so when I say remote sensing, I'm referring to aerial and satellite imagery. And what we can do is we can actually measure the wavelengths of reflected light across the electromagnetic spectrum, and that will tell us something about the health of the vegetation, how much vegetation there is, how much soil cover there is. And we can retrieve these different landscape traits from the satellite imagery. And importantly, we can reconstruct the past by going into satellite imagery archives. And this works really well for some traits. Again, vegetation cover works really well. You can get really nice indices of vegetation health, but it doesn't work for everything. And so there's only certain traits that can be detected reliably from remote sensing data. And so in this study, we combined field surveys and remote sensing. We recognize that they each have complementary strengths, and we kind of put them together in order to assess post-fire recovery during drought conditions. And so our field surveys, we did 82 belt transects within the burn scar throughout Los Padres National Forest. The transects were 45 by 5 meter rectangles, and we had field technicians do the typical demographic measurements in each transect, recording the species that were present. And then we also used close-range digital photography to estimate the cover of vegetation within each transect, and this was a measure that we could relate to the satellite imagery. And so what we did is we went down the transect tape, and we had a digital camera mounted on a pole, and we took kind of top-down images of each vegetation stand, but we did it, while standing right next to it, which gave us really reliable data, and we could analyze those photos in the laboratory and get really precise measurements of vegetation cover. And so pretty pictures time. This is me, walking down the transect tape for one of the transects that we did inside of the burn scar. This is what the digital camera setup looked like. And so that's an extending PVC pole. We have a camera bracket mounted on top. And so we were able to walk down the transect tape, and we did, I think, 8 photos down each transect. And then we can take those photos back to the lab and get really reliable information about vegetation cover, because the photos themselves, were very nice, very close up. And then we could also cross-reference with our field notes from our technicians, and we could even identify individual plants within each transect. And so it gave us a really kind of durable archive that we could go back and analyze later and cross-reference with the remote sensing imagery. For the remote sensing, we acquired satellite imagery from Landsat from 2000 to 2018, and we used that to monitor vegetation greenness trends. We used a technique called spectral mixture analysis, which is somewhat similar to NDVI, and we retrieved the fractional cover of green vegetation. And that's nice because it's a physical quantity. The fractional cover there actually gives you a % cover on the ground that you can relate to field measurements. And again, the fire occurred in 2007, and so that gave us 8 years of baseline data and then 12 years of post-fire data. And then Landsat has 30-meter pixels, and so this was kind of a stand-level snapshot within each pixel. And then what we did is we combined our two data sets. And so we had field survey data that gave us very reliable an understanding of where different species were in the landscape. And we had our remote sensing data where we could pick any pixel in the landscape and get a 20-year greenness trend. And so what we did is we overlaid those two data sets on top of each other. And then for each of the transects that we went out, we then had a 20-year kind of time series of what that health looked like over time. What we're able to do then is group those transects by dominant species and get kind of an average recovery trend for each species. And importantly, that revealed the impact of the 2012 to 2019 drought on post-fire recovery at an individual species level. And so by synthesizing the field survey data and the remote sensing, we're able to get more data, I think, than we would have gotten from either method individually. And conifers are also really interesting, but sometimes the closed canopy makes it difficult to do the field work. So we used a slightly different protocol for conifer forests. We extracted stand locations from CalVeg, which is a Forest Service database, and then we manually classified the pre-fire and post-fire cover using aerial imagery. And then we did the same thing where we overlaid that on top of our greenness data, and then we extracted trends for each unique combination of pre-fire and post-fire canopy cover. So next I'm going to talk a little bit about the results that we got out of this analysis. So here you see the greenness trends that we got for some dominant species. On the x-axis you have year running from 2000 to 2018. On the y-axis you have green vegetation fraction, which is basically the percent cover of green vegetation within each stand. So a value of 0.75 indicates that there's 75% green vegetation cover. What you can see is that before the fire, it changed a little bit year to year, but there's about 75% green vegetation cover for these species. Then hopefully it's pretty obvious where the fire took place. In 2007, the greenness drops to 0 for pretty much everything. And then from there, the race is off for the recovery process. But what we see critically is that there are different recovery trends for different species. And so for whatever reason, Ceanothus leucodermis seems to be a little slow off the gun, and it was a year behind everything else recovering. On the other hand, Stercocarpus speculoides, which is the darker purple line, did fairly well and seems to be pretty resilient throughout the time series. The other really critical trend here we see is that drought trend from 2012 to about 2018. We can see very clearly in this data that the drought delayed the post-fire recovery and actually set back the recovery process by several years. So this is really crucial data, which suggests that you had the initial disturbance of the wildfire, things were growing great, but then once you get that second disturbance of the drought really slowed that process down. And in a lot of cases, we see that the vegetation hadn't recovered to pre-fire conditions, within that 10-year time window. We did something similar for the conifer forests. Again, we grouped the conifer stands based on both their pre-fire and their post-fire canopy cover. And so this on the top right is kind of your best case scenario here. This is where you have dense canopy before the fire, that's the tab on top, and then you have dense canopy after the fire, which is the tab on the right. And here you see it's a pretty flat trend. It takes a bit of a bump when the fire happens, but in general it still seems to be a fairly healthy stand with high levels of vegetation cover. This is your worst case scenario down here. This is dense canopy before the fire and no trees at all after the fire. So this is a complete loss of what was a very thriving and dense conifer stand. You can see extremely high vegetation cover before the fire, plummets to 0, and then the post-fire recovery doesn't even get close to pre-fire conditions. And in fact, because there's no conifers after the fire, that's probably things like shrubs and grasses filling in. So it's not even the same species that we see there. And so using this method, we can kind of figure out what those trajectories would look like based on different pre-fire and post-fire outcomes. We also tabulated the number of conifer stands that had the unique combinations of pre-fire and post-fire conditions. So that right bar there is all the stands that had dense canopy before the fire. There's about 275 stands. And you can see only 139 of them still had dense canopy after the fire. And so we see really widespread loss of the Sky Island conifer forests. And in some cases, the red bars, you can see that there was complete loss of those conifer stands, which is probably type converted to some other sort of chaparral. Anecdotally, I can tell you that there are places where entire hill slopes of conifers were lost within the burn scar. And a lot of the vegetation coming in after was often Ceanothus palmeri, which seems to be fairly effective at outcompeting the conifer seedlings. And there were similar observations after the cedar fire in the early 2000s, so it might be interesting to examine Ceanothus palmeri in a little more detail, looking at its interaction with these Sky Island forests. And this is what one of those stands looked like. This is on top of Big Pine Mountain, which is kind of the very highest part of Los Padres National Forest. So you can see here that there was a conifer canopy that was probably intact previously, fairly dense canopy. It's almost a complete loss now, and you just see the conifer skeletons. The shrubs below there are Ceanothus palmeri coming in behind. You see a few remnant trees around the fringes, but by and large, we see complete loss of the conifer stand in this particular area, and we saw this on other areas of the landscape as well. So just for a few discussion points, we observed that the 2012 to 2019 drought really set back the post-fire recovery. And in some cases, we don't see stands recovering to pre-fire conditions. We saw that the individual chaparral and oak species exhibited distinct recovery trajectories, potentially due both to their post-fire behavior and then also their drought tolerance as well. We see that many conifer stands lost canopy cover, and in some locations, entire hill slopes of conifers were lost. And we think that remote sensing and field surveys provide really complementary approaches to examine post-fire recovery, where you get the certainty of a crew of field technicians with the spatial and the temporal scaling provided by satellite imagery. With that, I'm happy to take any questions. We've written this up in an article that's already been published, so if you'd like to read the article, it's open access. So you can use that QR code there. We'll take you to the article. If you have any questions, feel free to email me, and happy to take any questions with any time remaining. Thank you.
Are there any questions? I've got one in the back and then a couple in the middle part here. So.
When you're measuring the greenness, how did you account for any seasonal variation? Did you make sure that you had your remote sensing data collected at the same seasonal time of year?
Yeah, that's what we did. We did peak phenology for every year. So to a certain extent, there's interannual variability just because of interannual rainfall differences, but we did peak phenology every year, at least to control for the phenology.
Okay, got it.
So is CNPS going to use some of those tools for your next mapping?
We'll see. Yeah, we'll see. That's not a question for me.
Yeah.
What software do you use for your spectral mixture analysis?
We use ENVI. And then we have another tool called Viper Tools, which is a free extension for ENVI, but ENVI itself is commercial. Yeah, Viper Tools. Yeah.
So with your the photo transects,
Yeah.
do you use the same software to analyze and recover?
We used an open source software that was developed at Wisconsin, and what it does is it basically randomly samples pixels within each image, and then the pixels are manually labeled by technicians. And so I think we did something in the realm of like 200 pixels within each image, and that gave us kind of a representative sample of vegetation cover. So it's a method that could be replicated fairly easily. The specific code we used came from Wisconsin, but it's an easily transferable method. Yeah. Tell the Forest Service. I think there are. I think from what I've heard, the Forest Service does want to do follow-up studies. I'm not personally involved in that, but I think UCSB as a whole has been pretty involved in that effort. So.
Did you run into parallax error with the digital validation photos?
Everything was ortho-corrected. Yeah. NASA does all of that beforehand. So. Actually, USGS does Landsat, so USGS does it.
I meant about the digital photos taken on the floor.
No, we did our best in the field to kind of manually validate that it was flat, but we didn't do any more complex corrections of that. Could be good future area of research, I think, if we want to keep doing this. Yeah.
It's interesting in the baseline data, like all the species tracked the same year, and sorry, in the greenness graph.
Yeah.
And why is that? I wouldn't expect all species to.
That's your interannual variability in rainfall. And so the years where they're lower, it's just a slightly drier year, and the years where they're higher is
That they all.
Yeah.
Think that just because it's a little drier, every single species drops.
Yeah, it was interesting, yeah. And so, yeah, if you look at that pre-fire, all the variability you see there is that interannual in rainfall. And then everything post-fire is some combination of like seeding, sprouting, drought tolerance, stuff like that. So.
I had one more question.
Sure.
Is there a look at the survey of the 30s to do a little bit of comparative in the same kind of footprint because they did field work and mapping to...
Yeah, we did a little bit of that. With the 30 surveys, it can be hard to make sure you're in like the exact same spot. And so we didn't ultimately pursue that just because we had, a limited grant period. But I think that would be of interesting, if we want to assemble these really long-term comparisons. So.
All right, I think we gotta move on, sorry. Thank you.
Thank you.
All right, we've got a great group of people that are going to give the next talk. Okay, sorry. Oh, geez. Wanted to make sure I got the right place. So we've got, I think all four of you here, right? Yeah, great. Okay, we have Michael Pazmino, Minerva, Lara, Joshua Moreno, and Sebastian Alvarez Espinoza, and they are going to give a talk about their work as graduate students at CSU Long Beach's GIS program, where Michael has a background in visual communication at Cal State Fullerton. Minerva specializes in biology and plant identification. Joshua has studied geography at CSU Long Beach, and Sebastian is from North Carolina and has a background in geology, and it's a really interesting project that they all have come together that they'll tell us about.
Hello, everyone. Good morning. So we're going to start out with Sebastian. He's going to give us a quick overview of the study area. So Sebastian, go ahead and take over.
How's everyone doing? I hope everyone's awake. I've had enough coffee in your... So we're going to start with the start of our show, the Palos Verdes blue butterfly. It's a very unique, very rare butterfly. It's very majestic. It's known for its very bright blue colors and silver. It was thought to be extinct in the 1980s. It was not seen at all until it was found back in the 1990s. This is like one of the main reasons why the Palos Verdes Peninsula Conservancy was started, because a lot of people were seeing all these native species, native plants, like just being wiped out because of overdevelopment. And the Conservancy right now has 1,600 acres, and every year they're trying to acquire more land from, orange farms or private lands, and they've done a good job at it. So some of the species that we have over at Palos Verdes Peninsula are the coastal gnatcatcher. This bird is very native to coastal sagebrush, and its population has been wiped out. Its region goes all the way from Southern California all the way to Baja California, and talking to residents everywhere, they see like there's less population of them, less migration. Also LA has like a lot of noise, and the noise, the planes, the cars, the pollution is not the best environment for them. So, and the cactus wren is another one of our species that has been wiped out, slowly from almost every part of our urban region. So the Palos Verdes Peninsula consists of four different municipalities: the Rancho Palos Verdes, Rolling Hills in the Rolling Hills State, and San Pedro, which is a community that's part of Los Angeles. Overall, we've had this very big area that we like to map out. We wish we could put our drone through the whole peninsula, but as you know, it's against the law, so we've had to pick very unique places to run our drone, and had to get help from our professors, because I don't think we can afford a $50,000 drone in the water. And as you see here, and you probably know that Southern California is very well known for its wildfires. Every year we were expecting wildfires, but now it's not a few months, it's just half the year where we're seeing it very common. It's just a common sight all the time. I moved from North Carolina to California, and I didn't think it was that bad till everyone was telling me. And over over the years, there's been more acres burned, and especially in Palos Verdes Peninsula, where people lost their homes, firefighters have lost their lives, and this is a very small community, and the plants are very rare, the species are very rare. It's something we like to help and mitigate the efforts of the some fire departments from coming to destroy that land, because they might do it from fire mitigation, but it might not be good so much for the nature there. So going over to our ecological survey, we picked the Abalone Cove and Portuguese Point area. As you can see here, this is like it's a very big park on the right side and left side, and you can see on the right side is more it's more nature. There's some efforts to go in there to plant more native species, but we've had issues with the fire department, coming in and saying, well, we don't want we want the plants here, but if they're fire, it just might start fire because we don't get any water, So, we had one rain last week, and that's the first time I've seen it in months. So, over time, we wanted to basically go and fly some drones and see what we could find and see if there's any patterns we could see that we could help in, decide on how we can go about planting more plants and, finding any more species. Now I'm gonna pass it off to Josh here.
Hey everyone, so my section would be the pre-flight preparations and what we did to conduct the aerial imagery. So as you can see here, this is just a methodology flowchart encompassing all the steps that we took to complete this project, starting from creating our data dictionary to pre-flight preparation, the imagery analysis, and then our final presentation. So moving on to the pre-flight preparation, we use a drone deploy app to establish 3 study areas. So the park area is split in 2 sections, on the first 2 green ones, and then the third section is Portuguese Point. We did have a 4th one, and we did fly that part of the park, but ultimately we didn't conduct any surveying there. So the flight was conducted January 11th, and that was our second try at trying to fly because of the conditions. The first time were really poor. So yeah, in terms of the date, we chose January because the bird nesting began in February, so we had to choose just a date before that. And so what we used provided by our department was the DJI Matrix 200 quadcopter with the MicaSense 10-band imagery camera rig. And then from there, we basically put down ground control targets throughout the study area just to geo-reference the imagery with the ground control points. And for that, we use the EOS Aero 100 GNSS receiver, also provided by the department. And then here is a short video of the drone taking off. If it plays, it's fine. So image there is Scott Winslow. He's our department pilot and professor. He helped us throughout this entire project, and without him, this couldn't have been done. Oh, there it goes. Yeah, so it was an awesome piece of equipment to use throughout the entire project, and yeah. So
That's Minerva.
that's Minerva. Okay.
Minerva's going to talk about post-flight.
Hello. So this is the part after we flew it, we had to go on the field. So when we were on the field, we went and we took our EROs, and so we looked at the plants. And depending the size of the plants, we either small little buckwheats, we would put them in the center, and we would basically calibrate them for the accuracy using to connect to the field maps. But plants like lingonberry and all those, if we didn't have enough time, we would just go around them, to get the accuracy, or we would be close to them in the vicinity and then kind of map them ourselves to get the data. So we did do some hiking, and it was safe, but it was interesting. So the data, there was a couple of clips where we were like, okay, maybe not. So this is pretty much using the aerial image that we collected using the UAV. We also, like I said, took the polygons. Usually you could see them a little better, but I think it's a little hard to see from there. There should be like little red circles. They're there, but I don't know if you guys can all see them. They're like there. So those specify where we found specific plants, and we are using this specifically to help us dictate where the natives and the invasives are found. So one of them was Abalone Cove Park, and the other one was Portuguese Point. I believe it was a total of 150 data points, but let's keep in mind that this imagery was taken in January. So when we went out to do the field work, it was already March because the time went by. So when we went on the field, it was kind of different because by this time, like, you could see that there is some like very, very potent, like lemon berries, like they're very strong. But at the same time, when we went on the field, the California Encilias, they were just, we couldn't really see them anymore because they were like, their time is gone. But kind of more into that. So when we went in the field, specifically with the assistance of the bio from Palos Verdes, Austin Parker, and myself, we went on the field and we created a field dictionary that pretty much listed like each species that would probably be found. There was like a total of 66, but we like broke it down by a lot. So basically, we stated that this is the plant that we're going to be looking for specifically, and this is how to tell its features based on the leaves, the white flowers, or the big bush. Like, we would kind of go from there, and we would do the ground truthing, and I believe it was pretty successful because the group was able to do it like so successfully. Like, because at first they were like, "What plant is this? What plant is this?" And then I was like, "Lemonberry, lemonberry." but after a while, like, it was really good. After the field dictionary, they were able to do it on their own without like, and it was amazing. So these are kind of the plants, like I said, that we were looking for. So again, out of like 66, we cut it down by the most dominant species that were present. So we broke it down, and then so most of the plants that we focused on were the dominant, like lemonade berry, California brittlebrush, and prickly pear. We did see a lot of invasives. Like I said, the time has passed, especially for mustard. It was really hard to map. But we focused mostly on like Brazilian pepper trees and pine trees. But so after collecting all the data, and we figured out that the flammability at-risk species, we figured that out of like all the plants that we collected, that it is pretty certain that most of the plants that are going to be harmed by wildfires could potentially be California gnatcatcher due to like invasive species, or just the wildfires, because wildfires aren't necessarily like horrible because a lot of plants need them to regrow. But if invasive species keep coming, then potentially they won't come back. So Michael will explain more about our analysis.
All right, so we chose, let me go here. So we chose two different algorithms to classify our results. So we used the maximum likelihood classification, which is a supervised pixel-based classification. And then we compared that against a support machine vector algorithm using object-based classification. The first one, this is, we did all of our analysis through Envy. So we created this image with a combination of Envy and ArcGIS Pro. In the box A, you'll see invasive species kind of contaminating that area. It's actually the area where they were planting a new native plant species, and so that's like their little nursery. But then we started to see some invasives there as well. In yellow, you'll see California Encilla, and the dark green is the lemonade berry, and then the light green are the rest of the native plant species that we grouped up together as one group. So in B, you can see California Encilla and the lemonade berry. So one thing about the this classification is that although the native plant species have the highest spectral pixel count, as you can see in the bottom box, at, let's see, what did that say? Like 3,051,000 something pixels, they still had the lowest accuracy at 49%. And we'll talk about that, why that's happening, but there are issues to that. So when we move on to the Portuguese Point area, the trend is the same. Native plant species are at 20% with the highest spectral pixel count. And so in box A, we'll see California boxthorn, which has a very similar spectral index as lemonade berry. And so because of that, we'll start seeing it contaminate areas where the lemonade berry actually is. So in B, you'll see a little bit of like the red dots. And in that whole top left corner, you see a bunch of red and dark green, and that's because of the California boxthorn, which looks very similar to the lemonade berry. And so there's a lot of contamination with those clusters. And so one of the reasons why this might be is because we need to either further separate them into species level classifications, or there's just not enough data collected. Although we had about 120 something polygons, it's probably not enough for each of these areas. And so now when we compare them to the support vector machine algorithm, which is an object-based classification, we do have a little better results, and that's because the algorithm groups all of the similar pixels together and assumes that that's all the same species. But now, as we know, plants aren't exactly monogamous. They don't like to stay within each other. They like to live with other plants. And so the object-based classification may not be the best way to classify plants, whereas the pixel-based might be a little bit more accurate. And so now this is the Portuguese point. We have the same issue. Actually, this one in the box A, where we see the red, that is now the California boxthorn, the invasive species, and the support vector was able to classify that correctly because that area was identified as the invasive species. And then in the top left, as you see, there's more light green and more dark green. So in this case, they were able to classify that a little bit more accurately. And so what we did for the next steps in our future, we learned that next time for the next project, they'll have to fly at a lower altitude, focus on more significant plant species rather than attempting to collect all the species all at once. And then we had an interactive web app. So if you guys want to take a look at that, please take a look. Thank you so much. Oh, back a slide? Sure. Yes. I guess I could take one question. Anyone has a question?
Yeah, you got it.
Oh, yeah, cool.
Yeah.
Couple questions, sure.
Okay, does anyone have any questions? No one's taking a picture of your...
Taking a picture? Yes, in the blue.
Did you guys use all 10 bands for your classification?
Yes, we use all 10 bands for the classification, yeah. So we made a composite. So we first mosaiced everything, and then we composited all of the bands into a composite. Yeah, exactly. Yes, over there, sir.
From your experience with this, would you say that there's a acreage or just total area limit where you could do this kind of concentrated analysis and remote sensing before it becomes to a point where you're not able to click into it, apply it to an area far enough away? Like, is there a certain area that this works best at?
Oh, I see. Like if we should concentrate in a smaller area rather than the big park that we started at, or?
Well, no, I'm asking, do you have a sense of what the total area is that you could apply this effectively before you get too much
Oh, I see.
background?
I got it. Okay.
Screws up the results.
No.
You could apply it anywhere most. Your only enemy is time. Like if you fly at lower altitudes, just
Yeah.
go much longer. Also, weather. That area it's much like here in like the Bay Area. It's very cloudy, so sometimes it's, we would go out and we're like, oh, the fog is gonna settle.
So it can be applied to larger areas. The idea of our project was to it was a what's the word? It was a test. It was a pilot. Yeah, it was a pilot program that PVPLC wanted to use for the rest of their land. And so this area was 63 acres. The total is 1,600 acres. So this is just a pilot program. No pun intended with the UAV, but they want to see if this is even feasible. And so it is feasible, but like Sebastian said, we need more time and more resources. Yeah. Was there another question over here? I think I saw a hand. Yes. What do you recommend for your next survey effort? Yeah, that's a great question. Yeah, go ahead, Minerva.
I was saying that because we were introduced to our project late, like basically winter is when they told us, hey, you're assigned to this project, and we're like, cool. But I think it would be better if we can like do it maybe in fall, like talk about it earlier and possibly get started on it during fall, because I know that's when like the California brittle brush is kind of blooming. So you can just kind of go from there, and then because the lemonade berry is evergreen, so I think that would probably be the best approach, because I know the nesting season covers like February to like August, and our thing is done in August. So I would like suggest like getting started like September, October, November, and see what you can see there, because you'll probably see more than we saw in January and February. But I would suggest that.
Thanks, everyone. If you have more questions, feel free to ask them.
I'll show you the photo, yeah. I'm so sorry. Quick. Sorry. Go. Thanks so much. Thank you.
All right, thank you. Okay. Next, we have Claire Mallen, and Claire is a vegetation ecologist with Valley Water in Santa Clara. And sorry, I need my cheater readers or something. Geez, this morning, not enough sleep. Where she manages an invasive plant management program and maintenance and mitigation of restoration sites. The work she is presenting has been done while she was a research fellow with Valley Water and is in support of efforts to track ecosystem conditions excuse me, and to help make informed management decisions in the region.
Hello, good morning, everybody. I'm Claire. As Julie said, I'm a vegetation program specialist at Valley Water here in San Jose. We're formerly known as the Santa Clara Valley Water District. Today I'm going to talk about a study that I conducted as a fellow for the district from 2020 to 2021. This project was assisted by my former supervisor, Zoe Diggory, who unfortunately couldn't be here today. And I wanted to thank you all for having me. So first, since I know some of y'all are in the room, I wanted to thank folks over at the California Native Plant Society, the Golden Gate National Parks Conservancy, and the Santa Cruz Mountain Stewardship Network for their technical support throughout the project. I wanted also to thank our landowners who allowed us access to survey on their property. Some of these are listed here. In addition, we also gained access to private properties as well. So first, I wanted to touch briefly on using reference sites for restoration and mitigation planning. Reference sites are vegetation communities that serve as a model for your restoration or mitigation project. They're integral because they inform project design and can help overall in determining your evaluation or success criteria. They should be relatively pristine with low human impact, like social trails or trash. They should have high native species biodiversity. They should be structurally complex, and they should be regionally appropriate to your reference or restoration site. Some downsides to consider would be some of these conditions may not be replicable. They may not reflect the same stressors of your restoration site. Think about if there's a road by your restoration site and there's not one by your reference site, definitely something to think about. They do not always capture, single sites don't always capture the natural variability in that ecosystem, so it's important to think about using multiple reference sites. And it's also really important to think about restoring to reference conditions takes lots of time and effort to emulate. You know, these reference sites took decades to become the way they are, and your restoration site or mitigation site might only be monitored for 3 to 5 years. So that's also really important when thinking about using these highly pristine, high-quality reference sites. So the purpose of this study was to harness the pros and address the cons of reference sites to inform project design and identify performance criteria, to document the composition and conditions of Santa Clara County's most pristine native ecosystems, to collect enough samples of different vegetation types to capture variability and relationships, and to help make reliable and representative reference site information readily available for project design and evaluation. So to achieve our goals, we first had to identify the location of these high-quality ecosystems. So I first researched various publicly available geospatial data sets that could help identify areas that were impacted, that could impact ecological conditions. I ended up using 3 data sets and overlaid them to get this kind of really coarse overlay analysis here on the left. I used data sets like land use, distance to roads, and history of wildfire. Another data set that I wanted to use that just didn't work for our scale was invasive species cover from Calypsi or Calflora. I think if you're working on a much smaller scale, this data set could be really useful. So once the overlay was run, approximately 40% of the county was classified as good or very good, symbolized by the dark green and light greens. You can kind of see there on the hillsides where there's a lot of protected lands. About 40% was classified as moderate in the yellow, and then 20% was classified as fair or poor, kind of symbolized by the orange or red. You'll kind of notice the pattern. That's kind of our valley floor, our urbanized environment, and down like kind of going south, you'll see kind of those pockets of Morgan Hill and Gilroy. Oops. For these valley floor areas, we used existing data from the California Rapid Assessment Method for riparian and wetland ecosystems to find high-quality riparian vegetation communities on the valley floor. And then based on this overlay analysis, we reached out to those landowners that were kind of in those yellows, green, and greens to gain access. And then you'll see on the second map, this highlighted blue area is all the properties that we were granted access to survey on. And then the last step was to get out in the field and survey. So you'll see all the survey points we ended up with after 2 years of sampling. And I'll talk about our methods in a minute, but just when we were looking for reference sites in the field, we hiked and drove on previously established trails and roads, looking for stands of homogeneous vegetation with high species cover, high species biodiversity, and low non-native species cover. And we didn't survey any stand that had greater than 10% non-native tree cover, greater than 25% non-native shrub cover, and greater than 80% non-native herbaceous cover. So for our data collection methods, we used a modified version of the California Native Plant Society Vegetation Rapid Assessment. We believe that this method would be the best method to collect characteristic information, species information, and plot information, and it would allow us to collect a lot of samples in a short amount of time. I think this was really useful as well because it helped inform some of the Santa Clara County fine-scale vegetation mapping. And using this method allows other people to use this data in various different ways. So after sampling was complete, we compiled all of our reference sites into a Excel database that is publicly available on the Valley Water website. You can use it to sort for single or multiple reference sites. You can use various parameters to sort the data. You can sort it by vegetation alliance and vegetation groups. These are all parallel to the classification in the Manual of California Vegetation. You can sort by location, elevation, soil type, watershed, macrotopography, exposure, and steepness. And for this presentation, I just ran some basic summary statistics on our data. We collected data from around 416 data points, locations, reference sites. Here we just split it roughly up by watershed. You'll see the least sampled watersheds were the Alameda and the the San Lorenzo watersheds. That's because those are two watersheds that Valley Water doesn't service. And then our most sampled watersheds were from Coyote and Pajaro. I think that's because that's where a lot of our protected lands are in Santa Clara County. Next, we split it up by vegetation group. For the purpose to fit on the slide, we kind of clumped different groups into kind of like our own macro group, so these are not parallel to the MCV. This is just to kind of roughly show that most of our sampled groups were from riparian woodland and shrublands, California oak and bay forests, and California coastal scrub and chaparral. And then closely behind is our freshwater marsh and wet meadow. That's because most of our kind of our focus was on ecosystems that Valley Water frequently mitigates for, which is our riparian woodlands, our wetlands, and our oak woodlands. Oops. So for the purposes of this presentation, again, we did summary statistics on three groups. First is our evergreen and deciduous riparian forest and woodland. We see that most of the average elevation of these ecosystems is around 143 meters. I'll give you a minute to look at this table. It's got a lot of numbers, but I took our top 5 most frequent tree, shrub, and herbaceous species. You'll see the frequency percentage of how often these species were found in our plots, our average percent cover classes. All of our cover parameters were recorded as cover classes, so kind of these wider ranges. And then for our riparian ecosystems, we also added a relative elevation to the channel and a relative distance to the channel. And so these relative elevations show kind of where on the site in relation to the channel these species were found in. So I think this parameter would be really useful in kind of deciding plant layout. So I mean, it's kind of something that a lot of practitioners know, that your platanus, your sycamores should be a little bit closer to the channel and between 1 to 2 meters of the channel, and that your Quercus ags, your coast live oaks, should be a little higher up at 2 to 4 meters in elevation to the channel. And then next, we have our substrate and vegetation cover summary tables. So for our substrate tables, I think this would be useful in kind of determining your site conditions. So if you want to, so this evergreen and deciduous riparian forest and woodland group, it includes our sycamore oak woodlands and our valley oak riparian woodlands. And I think it's kind of important to say you want to restore a valley oak riparian woodland, you see kind of our substrate, our large rock in between 3% and your small rock is 10%. So if your restoration site is really rocky and you're hoping to establish a valley oak riparian, maybe you want to decide to switch up your plant palette for greater success. And you also see that litter is at that 60%. Maybe you want that leaf litter, maybe you want to kind of incorporate some clean wood chips to kind of serve that purpose of that leaf litter. And then our vegetation category covers, again, symbolized as cover classes, will help inform kind of inform your evaluation criteria. So you'll see that our total veg cover is somewhere in between that 50 to 75%. So if you're having success criteria that's higher than 75%, you might think about kind of lowering that to like within that range. And then I also think it's really important to think about kind of our cons of where your restoration site is located and your stressors. So these ecosystems are, you know, kind of your pristine ecosystems in the upper watersheds. And if your restoration site is on the valley floor with much higher stressors, maybe you kind of want to lower your total veg cover to kind of in that like lower 50 to 60% range. So next, I have our freshwater, coastal marsh, and wet meadow group. Kind of includes our spike rush alliances and our Carex and Juncus alliances. You'll see average elevation is 215 meters average. You'll see our 15 most frequent species were all herbaceous, kind of ranging in between natives and non-natives. You'll see our most one of our high-frequency plants that had the highest cover was Eleochris macrostachya. That's kind of an interesting plant because when we were in the field, we were noticing where we were seeing it. We were seeing a lot of it as this Eleochris wet meadow at Anderson Reservoir. Again, our substrate and vegetation category covers, you'll see that our highest substrate percent cover was in the litter. So I think probably thinking about when you're maintaining your site, kind of leaving that thatch in place to serve that for that nutrient cycling. And then our vegetation category covers, again, with that total veg cover of 50 to 75%. I'm thinking when you're establishing your performance criteria, kind of keeping that in mind with our total non-native vegetation in between 5 to 15, thinking about those stressors, again, that if you do have a site that has higher stresses on it, maybe thinking about having a higher allowance for your non-native species cover. And then our last example, we have our California oak and bay forests and woodlands. You'll see our top 5 tree, shrub, and herb species. You'll see if you do restoration work in the region, these are, you know, no-brainer species, pretty common species. I think it was pretty interesting that we found poison oak, toxicodendron in 90% of our plots. No shocker there, but I think it's kind of an indicator that this species is going to be kind of our powerhouse when we're thinking about making our sites climate resilient. I know it's not a fun plant to think about planting or maintaining in a restoration site, but definitely something to keep in mind. And lastly, we have our substrate and vegetation category covers. Again, leaf litter being a high percentage as 51, maybe want to think about incorporating a clean mulch there. And then for time, like I said, our database is available on the Valley Water website. The links here, you just have to go to our website, go to project updates under D5 ecological data collection and analysis. And thank you all for coming.
All right, we've got time for questions.
Awesome.
All right, we'll start close and work back, I guess.
So you mentioned early on when you were doing your suitability and your overlay, there was a layer available data set that you excluded on invasive species. You alluded to it as an issue of scale. So can you elaborate a little bit more on what the problem was and why you excluded it?
Yeah, so I didn't spend too much time on this data analysis. It was kind of just like a quick and dirty way to find these like high pristine areas and like to decide like where should I start looking. But when I did start looking at like data to download from CalIPSI or CalFlora, it just looked very overwhelming. Like, and you know, like not every population is recorded as well. So there's a lot of holes in those data sets as well. So just kind of decided.
We should talk.
Okay.
Some ideas and solutions. All right.
Yeah.
Okay. Rosie?
So you put this out there for restoration. Does Valley Water actually have expectations? Are they an organization that actually monitors restoration? And are you going to use that 5 to 15 and say, hey, 5 years, you're over 15%, that's a fail. You got to fix that? I'm just wondering how it's going to end up being used.
Yeah, of course. Yeah, so we're primarily a water supply and flood protection agency. So a lot of the projects we do are surrounding, like keeping our creeks clear. That requires removing vegetation, and then that requires us to mitigate for those impacts. So we do have a lot of riparian mitigation and restoration sites that do have really high success criteria. So we're hoping this could inform us to maybe we can lower those success criteria a little bit based on our overall reference conditions.
All right. A few more?
Yeah. Something you said earlier made me curious about the selection of reference sites. And while you're selecting the criteria of a site, are you selecting for healthy or resilient sites, or are you selecting for pristine sites, and why?
Yeah, that was definitely something we struggled with because a lot of where our restoration sites are on the valley floor, and it's kind of hard to make this connection of we have this super pristine ecosystem in the upper watershed. How do these sites, how do we rectify that? So as our study went on, we decided maybe we should lower our expectations and start to survey some of these more resilient sites in the urban core. So you'll see some of our sites ended up kind of being in that center on Coyote Creek and on Guadalupe Creek, trying to decide where are these nicer, or where do we have high species cover, a lot of native species, but within these stressed areas.
There are so many more questions. Oh, gosh. All right, Janelle, you're really standing there, but get going quick.
Hi, Janelle. No. They looked like our native sycamores, but not the hybrids are very more a little straight up, but yeah, we weren't able to do much.
Thanks. Make sure to catch Claire later if you have additional questions that she wasn't able to answer. Thank you, everyone. Thank you, Claire. All right. Last in this section of our two-part session, we have Danny Franco, who's going to speak, and he is a senior project manager working for the Golden Gate National Parks Conservancy, and excuse me. He's been working for the Conservancy for 12 years on natural resource conservation, vegetation management, and landscape scale habitat restoration projects. And he's also joined at the hip in this presentation by Mark Tuckman, who's the owner of Tuckman Geospatial, who uses state-of-the-art techniques to incorporate computer-based machine learning and aerial imagery and interpretation of vegetation, wildfire impacts, and land cover with using LiDAR and imagery. And then third, Cass Green is a collaborator with experience that spans over 30 years of managing and supervising GIS and remote sensing professionals for vegetation mapping, as well as leadership in GIS and remote sensing research and policy. So please come up. Danny, thank you.
Thank you, yes, and thanks to Mark Tuckman and Cass Green and a bunch of the other collaborators that I see in this room. Because my acknowledgements are at the end and I might not get there, Susan might tell me I run out of time. So for those of you in the room that helped me with my work and our work, thank you in advance. Yeah. So really quickly, the outline today, I'll give you an overview of the fine-scale vegetation mapping projects that have been unfolding in the Bay Area since 2013. We'll talk a little bit about the data products and methods. I'll probably go quickly through that and leave methods questions for cocktail hour or something tonight. How the data is being used, I think this is really interesting, and I'll spend some time on applications instead of methods, and then hopefully leave time for one or two questions. I'll hang out, and if you have questions, we can talk after the session. So the challenge that has inspired these projects around the Bay Area is that to effectively care for ecosystems and infrastructure, decision makers need to understand the location and distribution of resources across the landscape. Unfortunately, there are a lot of data gaps that exist across the state and regionally in the Bay Area. Existing fine-scale vegetation maps, this is not to pick on anyone who's involved with FVeg in the room, but sometimes they're a little bit coarse for local decision-making. In other cases, this is the Santa Clara Valley habitat map. It doesn't cover all the county, and so to do cross-jurisdictional work, these maps are insufficient. Similarly, topographic data is sometimes lacking and missing. And data such as these result in land managers sort of wrestling and laboring with the maps, arguing about which data sets to use instead of arguing about how we're going to solve problems. And that's what we need to be focusing on. So starting in 2013, we're going to talk about solutions. A group of agencies, NGOs, local, regional, state folks, some people in this room, came together to co-fund and implement countywide fine-scale vegetation mapping in Sonoma County. That started in 2013, was completed in 2017, led by Sonoma Ag and Open Space. They did fine-scale vegetation mapping pretty much to the California state standard held by CDFW, VegCamp, CNPS vegetation program. They also did carbon in Sonoma and fuels and other land cover mapping. Following in Sonoma's footsteps, the agency I work for, the Parks Conservancy, and our partners in Marin, the One-TAM Collaborative, initiated the Marin Countywide Fine-Scale Vegetation Map and Landscape Database in 2018. That was completed in 2021. Because my mentor Sharon Farrell likes to do things ambitiously, we decided to go ahead and do San Mateo County at the same time as Marin, which was a fun couple of years for me. We built a consortium similar in that it was made up of NGOs, local government agencies, and other collaboratives, but a different consortium in San Mateo County to deliver that map in 2022. During that same period, because go big or go home, we were doing some fuels mapping in Napa County, and actually Elkhorn Slough was mapped to the enhanced life form scale, the group level more or less of the NVC MCV, and then of course the folks at the Santa Cruz Mountain Stewardship Network got wind of what was happening in San Mateo, became extremely jealous, and invited us to go ahead and just keep moving down south. So people like Brett Hall and Julie Evans and folks in this room we were all game to just keep it going, keep the train going. We were able to fund that work, and that's going to be completed in June of next year. So that's actively happening. And then at the same time, and this is how snowballs happen, right? East Bay Regional Parks folks, so Alameda, Contra Costa County, a lot of the land managers in Santa Clara County, including SFPUC, have lands that cross these artificial boundaries called county lines, and decided that they should get together and try to fund East Bay, Alameda, Contra Costa. That's happening now too. So this is a really exciting time to be in fine-scale vegetation mapping in the Bay Area. Very quickly, some product examples. These vary county by county, but the kind of the thread that we can say it extends through all of them is that they are of high enough resolution to support land management decisions and of sufficient consistency to allow for that regional analysis, right? We want these to work on two scales. So, of course, LiDAR foundational, QL1 is the best. We partner with the USGS 3D program. That's the 3D elevation program. They're fantastic. We need imagery to do this mapping, 3 to 6 inch. It's got to have that 4th band. We produce an enhanced life form map. It's a more coarse than a fine scale vegetation map. Think of conifers, deciduous hardwoods, evergreen hardwoods. It's about a 20 to 26 class map, a precursor to the fine scale vegetation map. In Marin County, we did this really exciting collaboration with the state steward of the NHD to use elevation derived hydrography to update the NHD and WBD. That was more complicated than I thought it would be and took several years, but we were able to accomplish that, which is a really exciting thing. First thing for the state of California is Marin County having that NHD elevation-derived hydrography integrated into the national data set. Cornerstone of the success for the fine-scale vegetation mapping piece is floristic classification to the state standard held by CDFW VegCamp, CNPS vegetation program. I use this report in Marin County almost more than I use the fine-scale vegetation map. The information in there is absolutely critical to understanding not just the distribution, but the composition of these vegetation communities. Wildland fuels, hot topic, pun intended. LiDAR-derived ladder fuels, thank you, everyone's still awake. LiDAR-derived ladder fuels, these are really interesting density of, when you think about LiDAR and vegetation mapping, the thing to understand is that LiDAR gives you that vegetation structure, right? So we're not just talking about what type of vegetation, what are the floristics of that stand, but now we're talking about what is the structural composition of the stand. Ladder fuels, density of LiDAR returns in the 1 to 4 meter strata, topographic layers, you all have seen these. More vegetation structure, this is Potrero Meadow on Mount Tamalpais, and I just love looking at tree crowns in like this crazy X-ray way that LiDAR gives. It's just amazing, right? So many applications, the pervious and impervious surfaces mapping distance to roads, trails, so that you know where your weed pressures are, so that we can manage expectations for our reference conditions, right, Claire? Fine scale veg map, here it is, the kind of crown jewel of the effort, 100 plus classes, generally to the alliance level, although as you get down to the herbaceous, it's going to be much more coarse. We've been including tidal wetlands in most of these counties. And Cass Green, who's, as Julie said, joined at my hip, would tell me right now to make sure I say that all of this data is meaningless if you give people no access to it. You must give people access to the data. And so we built a project data portal. Thank you to CAL FIRE Santa Cruz Mountain Stewardship Network for helping us enhance this data portal, pacificvegmap.org. You guys can all have these slides when I'm done. Methods, we're going to go really fast. LiDAR is getting cheaper. Imagery is getting cheaper. That makes this more feasible. The coordination, I've said it a few times now, but I'll say it again, with CNPS Veg program and the VegCamp folks, these are the folks that hold the standards so that the mappers don't just go willy-nilly and make up their own classes, which they are prone to do because they can see it in the imagery. But it's really important to have these folks holding us to a standard because you get that ability to scale up and look at regional and then statewide trends, right? We don't want maps that only talk to themselves. That's just weird. Innovative technologies, that just means that Mark Tuckman knows how to use machine learning to make the machines do the easy part, like recognizing things like forest and grassland. And then, you involve humans to get to the fine scale, of course. Not mentioned, but should be, is aerial information systems when we talk about Tuckman Geospatial. They're the expert photo interpreters. The ability to make these products widely and easily accessible on the web builds a constituency for a wide user base. And the economies of scale from region-wide cross-jurisdictional collaboration. This is where I shine. I bug all these people. And then they give us money, and then we do the work. So thank you, everybody whose logo's on there. 3 primary objectives for methods, right, is understanding variation on the ground, understanding variation in the imagery and ancillary data, making connections between the variation on the ground, variation in the imagery and ancillary data. This is China Camp. The fine scale veg map did do pretty good just in that area right there. Not so much in the other tidal wetlands, but we're working on it. To understand, we go to the ground in a few different places. We do existing field data aggregation. We partner with people like Claire Mallen to get their data. Give us your data, Claire, so that we can understand the local vegetation community floristic composition patterns and support that classification analysis that CDFW and CNPS partner on. We go to the field again to do reconnaissance to support development of the floristic heuristic key to help to inform the local descriptions that the vegetation program produces. They go to the field again during the manual editing. That's where Santa Clara is at right now in their fine scale process. So the mappers are having a hard time distinguishing between types, so they go to the field to try and do additional calibration. Understanding variation in the imagery and ancillary data. Image analysis, we use it all. Stuff with high temporal resolution, thank you, Susan, high spatial resolution. The ancillary data that is used are things like hydrology, distance to streams, elevation, you can imagine. I won't go through this slide because it's more technical than I have time for, but suffice to say that it is supervised machine learning, meaning that guy right there watches the machine do all the work. Local expert review of final draft. This is really critical, and I think a step that we haven't shortchanged and I think is so vital. These maps need to get in the hands of people that are familiar with these vegetation communities on the local landscape so that they can go to the area that's really special to them and tell us how we screwed it all up and how we can fix it before we make it public. This is something Julie started the session with, and I think it's really important is to think about scale and how this data works on different scales. And so with that in mind, we'll talk about 3 example applications. Oh, this is the Brady Bunch slide. It's good for everything, fine scale vegetation mapping. 2017 Sonoma complex fires happen. It's a really profound event for land managers in the region, sort of a wake-up call, right? That fire getting into Santa Rosa was a big deal. They were catastrophic. They also presented a unique opportunity to use data like fine-scale vegetation maps to understand pre- and post-fire variables and fire behavior. Sonoma Ag and Open Space partnered with NASA to study the relationships between post-fire woody canopy condition, landscape characteristics such as weather, vegetation type, fuel loading, land use, and land management patterns. That all got written up in a CDFW Wildfire Journal, Special Edition 2020. There's a link to that paper by Green et al. In this slide. But interestingly, the fine-scale vegetation map for Sonoma was just completed months before this study. So it really gave them an opportunity to study the relationships between the fine-scale vegetation community class, the structure of the vegetation, and then canopy burn severity, right? It's canopy burn severity and that relationship to pre-fire land cover variables. So a fascinating paper, and I encourage you to read it. In Marin County, some work that I'm working on with the One TAM Collaborative is to develop a regional forest health strategy so we can get state money for wildfire resilience and use it for forest health. This is an area in Tamal Space State Park where our fine-scale vegetation map has Pinus muricata, bishop pine stands. These stands are hammered by pitch pine canker disease, and in some cases, it's also because they're old and senescing and they haven't had natural fire in a really long time. But in any event, we can add on to our fine-scale vegetation map mortality mapping that's looking for pixels that aren't, green, they're gray. We can quantify that, and then we can look at the distribution of mortality across the landscape. That's also happening for the Santa Cruz Mountains project concurrently. But then you layer on that LiDAR analysis to give you an idea of the seral state. This is nice because bishop pine is like an even age cohort that, serotinous species that is really easy to connect LiDAR and derived stand height and seral state. Seral state for redwood forest is a little bit more complex, but we're going there with a One TAM collaborative. So this is just another example of the applications that you can use to really kind of understand not just the vegetation type and cover, but sort of what's really going on with the resource, at least without having to go to ground and get poison oak, like Claire said. In San Mateo County, they did this 5-meter fuel model. This is the standard Scott and Bergen 40 fire fuel models that are developed for the Sierra Nevada, but we use them on the coast anyways. This is the 30-meter land fire. This is the 5-meter model, so I'll just go back and forth real quick. 5-meter model is better. That's the takeaway. Especially in the urban areas, which for some reason everyone's really interested in when you talk about fuel modeling. This becomes the underpinning for using that impervious surface and structure mapping to then look at distance to structures and relative wildfire hazard. So you get these really nice, basically carving up the landscape into hot, medium, cold. This gives people a really handy prioritization tool that works, again, at that local scale. How'd I do, Susan? Time's up? I'm good. Okay, hopefully I didn't go too fast, but that gives us more time for questions. Thank you.
untrophic samples?
Well, that's a really good question. Julie probably wouldn't let me get away with less than 300, I think is like a good ballpark. But we as many as possible. Yeah.
I mean, there's thousands of reconnaissance level where they polygonized an area to get reference data as well as the ground level classification data. So there's kind of two scales. And then afterwards, there was the accuracy assessment where they held back some of the data and then collected additional data. So.
Yeah. Go ahead. Yeah.
Hi, you're doing a lot of work. Awesome.
Thankfully, it's not all me. Look at all those names.
Is there any effort, though, to go back into some of these burned areas now?
Yeah, so that's a great question. Through the Santa Cruz Mountain Stewardship Network and CAL FIRE, we were able to get funding in partnership with the San Mateo RCD to study the CZU fire. So it's similar, using remote sense data to kind of look at pre and post conditions, but we're going to go to ground as well to Big Basin State Park and try to come up with a classification that's meaningful for understanding canopy damage in redwoods. So, and everyone's really curious to know how those redwoods are going to do in Big Basin. So there is some effort to go to ground with respect to these large-scale mapping efforts. Yeah. Yeah, I'm not sure if they've gone back to look at that data, but that's a great question. Yeah.
Anyone else? Yes.
Is there a plan to expand this to all of California, or is kind of once those counties?
There is a plan, and I didn't come up with it. It's actually, VegCamp has been working on this since wholeheartedly, I think, for a long time. And there is a statewide plan to do fine-scale vegetation mapping. I think we're just standing on their shoulders and trying to help close gaps by this, kind of bringing together public and private partnership to finish the map for the state.
It takes a lot of federal as well as a variety of state agency funding from CAL FIRE and USGS et al. So it's a lot of grant writing.
Yeah, it's grant writing and, email and nagging via email. Yeah.
And this is more of a comment to that point, and I think this is something that would be of interest to all states with all of this federal funding coming down the pipeline for resiliency, for fire. I don't know if you all have plans to kind of have a place on the site to talk about how you achieved the crossover of funding, how you kind of achieved that partnership, because I think that's a model that other states need to look at in addition to the actual analysis itself.
Yeah, that's a great point. Yeah. There's some resources through the California Landscape Stewardship Network. So how do you get collaboratives to collaborate? I'm happy to share more about that with you after the session. Yeah, it's a great resource. But yeah, leveraging the impact of partnerships would be Sharon Farrell, my mentor's way of framing that. Yes.
Wonderful. No others. Thank you, everyone, for coming.